My name is Emma Johnston, and I will be talking about how the aggregation and spread of a protein called alpha-synuclein can contribute to Parkinson's disease. As you likely know from previous videos, Parkinson's disease is primarily caused by the death of dopamine neurons in the substantia nigra, which is a structure in the midbrain. In order to begin to study how to prevent this cell death, we must first understand how these cells die. In this video, we will be analyzing one cause for dopamine neuron death in Parkinson's. Most Parkinson's cases are late adult onset and sporadic, but a small fraction of cases are familial. This fraction of familial cases indicates a genetic component to the disease pathology. In 1997, studies of some Italian familial cases of Parkinson's in the village of Contorsi Terme, as well as a few Greek familial cases, resulted in the identification of the first familial Parkinson's disease gene. This gene, which is inherited in an autosomal dominant fashion, encodes for the presynaptic protein alpha-synuclein. The function of alpha-synuclein is not fully understood, but some studies suggest that it plays a role in regulating neurotransmitter release. The protein is also a major component of Lewy bodies, which are intracellular inclusions, or aggregations of this protein inside neuronal cells, and are a defining pathological feature of Parkinson's disease. Protein aggregations are a hallmark of many neurodegenerative disorders, as we saw with Alzheimer's disease. So we've established that alpha-synuclein aggregation has an association with Parkinson's disease, but what exactly can cause this aggregation? We know that one option is a genetic mutation, as we saw in the 1997 familial studies. Commonly, this is just a single amino acid change, and the misfolding it causes promotes aggregation. Additionally, researchers found that increasing the copy number of alpha-synuclein, and therefore inducing overproduction of the protein, can also cause Lewy body pathology typical of Parkinson's disease. Now that we understand a little bit about how these Lewy bodies form, we have to analyze where they occur and if they spread. Postmortem analysis of Parkinson's patients suggests that the pathology follows a spatiotemporal sequence. In early stages of the disease, more alpha-synuclein aggregation is found in the brainstem. In late-stage patients, however, this pathology is widespread in the forebrain. The spatial trajectory of this analysis implies that aggregations may spread from neuron to neuron. Though they begin formation in the brainstem, they can be transferred from one neuron to the next, which causes this spatiotemporal sequence. Overall, we see a trajectory of aggregation, followed by cellular dysfunction, followed by cytotoxicity, which refers to when these aggregations spread and essentially infect nearby neurons, finally followed by eventual neurodegeneration. So, how have scientists supported this hypothesis that alpha-synuclein aggregates actually spread from neuron to neuron? The first study that prompted this hypothesis was of Parkinson's patients that received transplants of fetal dopamine neurons. Presumably, these neurons did not have mutated alpha-synuclein and therefore were not predisposed to aggregates. However, postmortem analysis of these patients' brains showed that the transplanted neurons also contained Lewy body pathology, suggesting that these aggregates spread from other cells. Another study done in mice suggested that endogenous alpha-synuclein, in other words, alpha-synuclein that is originally found in the organism in question, is actually essential for aggregates to form at all. This is similar to what is seen in prion diseases, in that the presence of an infectious protein makes the non-infectious conformation more prone to adopt the infectious pathology. Researchers had two groups of mice, wild-type and alpha-synuclein knockout mice. The knockout mice did not have endogenous alpha-synuclein. They injected both mice with alpha-synuclein fibrils, which are groups of the protein that have formed insoluble fibers. When injected into the brains of these mice, these fibrils caused aggregation in the wild-type mice, but were not able to induce aggregation in knockout mice. This supports the hypothesis that the presence of endogenous alpha-synuclein is necessary for aggregation formation. Overall, these experiments suggest two things. One, recruitment of endogenous alpha-synuclein is essential for the pathology in question, And 2, Parkinson's disease may involve cell-to-cell spread of pathogenic proteins, in particular pathogenic alpha-synuclein. To summarize, alpha-synuclein is a presynaptic protein that, when aggregated, forms Lewy bodies, which are a defining feature of Parkinson's disease. This aggregation can stem from overproduction of the protein or formation of a mutated protein. It is hypothesized that these aggregations travel from neuron to neuron, thus causing the spatiotemporal development of Parkinson's disease. Overall, looking at how alpha-synuclein aggregates and spreads can aid scientists in understanding the causes of cell death in Parkinson's, and that understanding can help inspire future treatment.